Hello everyone, this is Rutuja Kolle and welcome to my channel Rutuja's World of Ideas. This is my very first video. तो इस वीडियो में हम एक बेसिक आइडिया लेते हैं लाइक व्हाट इज़ माइक्रोबायोलॉजी, देन व्हाट वी स्टडी इन माइक्रोबायोलॉजी. सो लेट्स स्टार्ट. Is the scientific study of microorganisms, tiny life forms invisible to the naked eye, such as bacteria, viruses, fungi, archaea, protozoa, and algae. These organisms are everywhere inside our bodies, in soil, water, air, and extreme environments like hot springs and deep sea vents. While a few causes diseases, most are harmless or beneficial, driving digestion, nutrient cycling, food production, and many industrial processes. In this slide, we are introducing the fascinating field of microbiology, which is the scientific study of microorganisms. Microorganisms are tiny life forms that are invisible to the naked eyes. We cannot see them without the help of microscope, which is why this field became advanced only after the invention of microscope. Microorganisms include a wide variety of life forms such as bacteria, viruses, fungi, archaea, protozoa, and algae. Even though they are microscopic in size, their impact on our planet is enormous. They are present everywhere in soil, water, air, and even inside our own body. In fact, the human body contains trillions of microbes, especially in the gut, which help in digestion and maintain our health. Many people associated microorganisms only with diseases. Yes, some microbes are pathogenic and can cause infections. However, the majority of microorganisms are harmless or beneficial. For example, bacteria help in nitrogen fixation in soil, which supports plant growth. Fungi are important decomposer in the ecosystem. Microbes are also used in food production such as curd, bread, cheese, and fermented products. Interestingly, microorganisms can survive in extreme environments like hot springs, acidic lakes, and deep sea hydrothermal vents. These show their incredible adaptability. So, microbiology helps us understand this hidden microscope world that plays a vital role in health, environment, agriculture, and industry. Without microorganisms, life on Earth would not be function properly. That is why microbiology is considered one of the most important branch of biological sciences. What microbiologists study? Microbiologists examine structure, function, classification, genetic ecology, and interaction of microbes. Tools include light and electron microscope, culturing techniques, training methods, example gram stain, and genetic sequencing. 16S RNA metagenomics. Organisms fall into prokaryotes like bacteria, archaea, eukaryotes like fungi, protozoa, algae, and a cellular agent like viruses. Recent advances emphasize culture independent methods to study the vast uncultured majority. Now let's understand what microbiologists actually study in detail. Microbiologists examine the structure of microorganisms, meaning their shape, size, and cellular organization. They study whether a microbe is prokaryotic or eukaryotic, whether it has a nucleus, cell wall, flagella, or other structural features. They also study the function of microorganisms. This includes how microbes obtain energy, reproduce, metabolize nutrients, and survive in different environmental conditions. Genetics is another important area where scientists analyze microbial DNA, mutation, gene expression, and evolution. Classification is also a key aspect. Microorganisms are grouped into three major categories: prokaryote, eukaryote, and a cellular agent. In prokaryote means including bacteria and archaea, eukaryotes including fungi, protozoa, and algae, and a cellular agent, mainly viruses. Microbiologists also study microbial ecology, which means how microorganisms interact with each other and with larger organisms like plants, animals, and humans. To study microbes, various laboratory techniques are used. These include light microscopy and electron microscopy for visualization. Culturing techniques are used to grow microbes in laboratory media. Staining methods such as gram staining help differentiate bacterial types. Then, modern techniques like 16S RNA sequencing and metagenomics allow scientists to study microbes without even growing them in a lab. Recent advancement focus on culture independent method because many microorganisms cannot be grown artificially. These techniques help scientists understand the vast uncultured microbial diversity present in nature. So overall, microbiologists explore the structure, function, genetics, classification and ecological importance of microorganisms, helping us understand life at the microscopic. Brief history from Leeuwenhoek to genomes. The field began when Antoine van Leeuwenhoek first observed animalcules in the 1670s. The 19th century saw Pasteur disprove spontaneous generation and Koch establish germ theory and pure culture techniques. In 1977, Carl Woese used rRNA sequences to identify archaea as a separate domain. The 20th and 21st centuries added molecular tools, sequencing, and CRISPR, transforming microbiology into a genomic and systems science. In this slide, we are looking at the brief history of microbiology. How this field developed from simple observations to advanced genome science. The journey of microbiology began in the 1670s with Antony van Leeuwenhoek, who is known as the father of microbiology. Using his handmade microscope, he was the first person to observe tiny living organisms, which he called animalcules. This was the first time humans realized that an invisible microscopic world actually existed. Later in 19th century, a major scientific breakthrough happened. Louis Pasteur disproved the theory of spontaneous generation and proved that microorganisms come from pre-existing life. He also contributed to pasteurization and vaccine development. Around the same time, Robert Koch established the germ theory of diseases and developed pure culture techniques. Koch postulates help scientists link specific microbes to specific diseases. In 1977, Carl Woese made a revolutionary discovery by studying ribosomal rRNA sequences. He identified archaea as a completely separate domain of life, differentiate from bacteria. This changed our understanding of evolution and classification. In 20th and 21st centuries, microbiology moved into the molecular biology. In the 20th and 21st centuries, microbiologists moved into the molecular era. Scientists began using DNA sequencing, molecular biology tools, and genome analysis. Techniques like whole genome sequencing and CRISPR gene editing transform microbiology into a genomic and system-based science. So, from simple microscope to advanced genome sequencing, microbiology has evolved tremendously. These histories show how curiosity, technology, and scientific thinking together shape our understanding of the microbial world. Gram stain and visualizing bacteria. The gram stain differentiate bacteria by cell wall structure. Gram-positive bacteria retain crystal violet and appears purple, while gram-negative bacteria take up counter stain and appear pink or red in color. Microscopy and staining remains foundational for classification, diagnosis, and guiding initial treatment decisions in clinical microbiology. Now let's talk about one of the most important techniques in microbiology, the gram stain. The gram staining method is used to differentiate bacteria based on their cell wall structure. It was developed by Hans Christian Gram in 1884 and is still widely used today in clinical microbiology. In this method, bacteria are first stained with crystal violet, then treated with iodine, followed by alcohol for decolorization, and finally counterstained with safranin. Based on how bacteria respond to this staining process, they are divided into two main groups. Gram-positive bacteria retain the crystal violet stain and appears purple under the microscope. This is because they have a thick peptidoglycan layer in their cell wall. Gram-negative bacteria do not retain crystal violet after alcohol treatment. Instead, they take up the counter stain and appears pink or red. This is due to their thinner peptidoglycan layer and the presence of an outer membrane. Gram staining is extremely important in diagnosis. It helps doctors quickly identify the type of bacteria causing infection and guides initial antibiotic treatment decisions. Even though modern molecular techniques exist, microscopy and staining remain foundational tool in microbiology. They are quick, cost effective, and essential for preliminary identification in laboratories. So, gram staining is not just a simple coloring technique, it is a powerful diagnostic and classification tool in microbiology. Core groups of microorganisms. First is bacteria. Bacteria are single cell prokaryotes 0.5 to 5 micrometer in size with varied shapes and cell walls that determines gram positive or gram negative staining. Roles of bacteria are gut flora, nitrogen fixation, fermentation, and some pathogens. Then second is archaea. It also pronounced as archaea. Prokaryotes adapted to extreme means thermophilus, halophilus. Distinct biochemistry from bacteria, important in methane production and environmental cycles. No known human pathogens. Fungi. Eukaryotes with chitin cell walls, yeast unicellular and molds multicellular. Important in food like bread, beer, antibiotics decomposition and some infections. Fourth one is protozoa and algae. Protozoa are often motile, single cell eukaryotes. Algae are photosynthetic eukaryotes that support oxygen production and food webs. Last is viruses. Cellular agents 70 to 1200 nanometer in size that replicate only inside host cells. Central to virology, vaccine development, and emerging therapies such as phage therapy. In this slide, we are discussing the core groups of microorganisms. First is bacteria. These are single cell prokaryotic organisms usually ranging from 0.5 to 5 micrometer in size. They have varied shapes like cocci, bacilli, and spirilla. Their cell wall structure determines whether they are gram positive or gram negative. Bacteria play important role in nitrogen fixation, fermentation, gut health, and some also causes diseases. Next is archaea. These are also prokaryotes but are genetically and biochemically different from bacteria. Many archaea are extremophiles, meaning they survive in extreme environments such as hot springs or high salt areas. They are important in methane production and environmental nutrient cycles. Then we have fungi, which are eukaryotic organisms with chitin in their cell walls. They include unicellular yeast and multicellular molds. Fungi are important in food production like bread and beer, antibiotic production and decomposition. Some fungi can also cause infections. Next is protozoa and algae. Next are protozoa and algae. Protozoa are single-celled, often motile eukaryotes, and some are pathogenic. Algae are photosynthetic eukaryotes that contribute significantly to oxygen production and aquatic food chain. Finally, viruses are cellular agents that can replicate only inside a host cell. They are central to virology, vaccines development, and new therapies such as phage therapy. Together, these groups form the foundation of microbiology and represents the incredible diversity of microbial world. Next is modern tools and breakthrough. In this slide, we are discussing modern tools and breakthroughs that have transformed microbiology into a highly transformed microbiology.
In this slide, we are discussing modern tools and breakthroughs that have transformed microbiology into a highly advanced scientific field. Earlier, microbiology mainly depended on microscopy and culturing techniques, but today the field has expanded into molecular biology and genomics. One of the most important tools is DNA sequencing, which allows scientists to read the genetic material of microorganisms. With whole genome sequencing, we can understand microbial evolution, antibiotic resistance, and virulence factor in detail. Another powerful technique is PCR, polymerase chain reaction, which helps amplify small amount of DNA for detection and analysis. PCR revolutionized diagnostic microbiology because it allows rapid identification of pathogens. Metagenomics is another breakthrough. It allows scientists to study microbial communities directly from environmental samples without culturing them. This helped discover the vast microbial dark matter, microorganisms that cannot grow in laboratory conditions. CRISPR-Cas system, originally discovered in bacteria as a defense mechanism against viruses, are now used for precise gene editing. This has application in genetic engineering, medicine, and biotechnology also. Advanced imaging techniques like electron microscopy provided detailed visualization of microbial structure. Automated culture system and bioinformatics tool have improved data analysis and research accuracy. These modern tools have transformed microbiology from simple observation to high resolution molecular science. Today, microbiology integrates genomics, bioinformatics, biotechnology, and system biology, making it one of the most dynamic and interdisciplinary scientific fields. Why microbiology matters? Everyday impacts. Microbes enable bread, cheese, beer, and yogurt through fermentation, produce antibiotics, penicillin, and biotechnological products such as insulin in ecosystem. They recycle carbon and nutrients, support plant growth via symbiosis, and help clean pollutants. In health, a balanced microbiome supports digestion, immunity, and even mental well-being. This slide explains why microbiology is important in our daily life. Microorganisms play a crucial role in food production. For example, yeast is used in bread fermentation, bacteria are involved in yogurt and cheese production, and microbes help produce beverages like beer and wine. Without microorganisms, many fermented foods would not exist. In medicine, microbes are used to produce antibiotics such as penicillin. Biotechnology industries use bacteria to produce insulin and other therapeutic proteins. Vaccines are also developed using microbial research. In the environment, microorganisms are essential for nutrient cycling. They decompose organic matter and recycle carbon, nitrogen, and other essential elements. Nitrogen fixing bacteria help improve soil fertility, supporting agriculture. Microbes also help in bioremediation, which means cleaning environmental pollutants such as oil spills and toxic waste. In agriculture, plant microbe interaction improve crop productivity. Inside our body, the human microbiome supports digestion, immunity, and even mental health also. A balanced microbiome is essential for overall well-being, so microbiology is not limited to laboratories. It directly affects food, health, environment, and industry. It is deeply connected to our everyday life. Branches of microbiology. Microbiology is a broad with specialized branches focused on organisms, environment, or applications. The table below summarizes major branches, their focus, and key applications. First branch, bacteriology, primary focus, morphology, physiology, genetics of bacteria, key applications and example, disease diagnosis, antibiotic and bioremediation. Second branch, virology, primary focus, virus structure, replication, host interaction. Key application and example: vaccines for polio, COVID-19 and phage therapy. Third branch, mycology. Primary focus on fungi taxonomy, ecology and pathogenesis. Key applications and example: antifungals, fermentation and biofuels. Next branch, phycology. Algae physiology and classification. Key applications and examples are aquaculture, biofuel production and oxygen cycling. Next branch is parasitology. Primary focus on parasite life cycle like protozoa and helminths. Key applications and examples are malaria control, tropical disease management. Then immunology, host immune response to microbes. Applications: vaccines development, allergy and autoimmune research. Then next branch is industrial and food microbiology. Primary focus on microbes in manufacturing and food, and applications are yogurt, cheese, enzymes, antibiotic production. Next branch, environmental and microbial ecology. Primary focus, microbe environment interactions, and applications, bioremediation, nutrient cycling, climate models. Last branch, microbial genetics. Primary focus, gene expression, horizontal transfer, and applications are synthetic biology, genetic engineering. Microbiology is a broad field with many specialized branches. Bacteriology focuses on bacteria, their structure, physiology, and genetics. It plays a key role in disease diagnosis and antibiotic development. Virology studies viruses, including their structure, replication, and host interaction. It is crucial for vaccine development and understanding viral diseases like COVID-19. Mycology deals with fungi, their classification, ecology, pathogenicity. It contributes to antifungal drug development and fermentation industries. Phycology studies algae, which are important for oxygen production and aquatic ecosystems. Then parasitology focuses on parasites such as protozoa and helminths, helping control diseases like malaria. Immunology studies the immune response to microbes essential for vaccine research. Industrial and food microbiology involves using microbes in food production, enzyme production, and pharmaceuticals. Environmental microbiology studies microbial interaction in ecosystem, including bioremediation and climate impact. Microbial genetics focuses on gene expression, mutation, and genetic engineering. Each branch contributes uniquely to health, industry, agriculture, and environmental sustainability. Challenges and future directions. Key challenges including antimicrobial resistance, undiscovered microbial diversity, and translating lab discoveries into safe, scalable solutions. Future directions emphasize metagenomic exploration of the microbial dark matter, phage therapy as an antibiotic alternative, microbial engineering for sustainable chemicals, and leveraging microbes in climate solutions such as carbon sequestration. Interdisciplinary research and stewardship are essential. Despite many advancements, microbiology faces several challenges also. One major challenge is antimicrobial resistance. Many bacteria are becoming resistant to antibiotics, making infections harder to treat. This is a serious global health issue. Another challenge is the vast number of undiscovered microorganisms. Scientists estimate the most microbial species are still unknown. Exploring this microbial dark matter requires advanced metagenomic tools. Translating laboratory discoveries into practical and safe applications is also challenging. New therapies and technologies must undergo rigorous testing before clinical use. Future direction in microbiology include developing alternative treatments such as phage therapy, engineering microbes for sustainable chemical production, and using microbes in climate solutions like carbon sequestration. Interdisciplinary collaboration between microbiologists, chemists, engineers, and data scientists will be essential for solving global problems. Last, summary and resources. Microbiology in brief. Study of diverse microbes that shape health, industry, and environment. Most are beneficial for neutral or small friction cause diseases. Careers and impacts. Paths include clinical labs, research industry, environmental consulting, teaching, and science communication, all crucial for addressing in global health and sustainable challenges. Recommended reference: Britannica Microbiology Overview, Microbiology Society, American Society for Microbiology, Liber text for textbook and primers. In summary, microbiology is the study of diverse microorganisms that influence health, industry, and the environment. Most microbes are beneficial or neutral, and only a small fraction cause diseases. Throughout this presentation, we explored the history of microbiology, major groups of microorganisms, laboratory techniques, modern tools, branches, and future challenges. Microbiology is not just about studying tiny organisms. It is also understanding life at its most fundamental levels. It connects medicine, agriculture, biotechnology, and environmental science for further learning. Students can explore reliable scientific resources such as microbiology textbook, academic journals, and reputable science organizations. Continuous learning is important because microbiology is a rapidly evolving field. Career opportunities in microbiology include clinical laboratory work, research, pharmaceutical industries, environmental consulting, teaching, and science communication. Microbiologists play a crucial role in addressing global health and sustainability challenges. So, overall, microbiology is a powerful and impactful scientific discipline that shape our world in visible and invisible ways. So, fascinating hidden world of microorganisms. If you found this video helpful, please don't forget to like, share, and subscribe to my channel. And yes, this is my first YouTube video, so your support truly means a lot to me. Please encourage me by subscribing and sharing it with your friends. Your small support will motivate me to create more useful educational content for you. On this channel, I will be uploading simple and easy explanations of microbiology and other life science topics, especially for students who want concepts in a clear and understandable way. Also, if you want notes, PPTs, PDFs, and extra study materials, then make sure you join my WhatsApp group. The link is given in the description box below. In the group, I will share class notes, presentation slides, important questions, study material, and update about upcoming videos. So please support me on this new journey. Thank you so much for watching, and I will be see you in the next video. Until then, keep learning and keep growing. Thank you.